Hi, my name is Scott Kitchen. I'm a professor of medicine at the David Geffen School of Medicine at UCLA, and I'm representing CDR3 Therapeutics, which is a company that we have formed to spin off the technology that we founded at UCLA, that we discovered at UCLA, in hopes of commercializing it and bringing it to patients. So CDR3 is a gene therapy company, and what we're looking to do is capitalize on our breakthrough approach for treating cancers and chronic viral infections through the genetic reprogramming of T cells using either chimeric antigen receptors or CARs or T cell receptors or TCRs. So these are molecules on the surface of cells that allow these cells to recognize and kill either malignant cells or virally infected cells through killer T cells. So in this case, we reprogram killer T cells to specifically target and eliminate these cells. So current gene therapy implementations of this technology involves the reprogramming of peripheral blood T cells from patients. And this has been hugely successful in the oncology field, the cancer field, in treating liquid tumors, but not solid tumors or chronic viral infections. And these indications are vastly under studied and are vastly, and the technology does not work for them. So the reason for this is these blood T cells are heavily processed, and when they're put back in the body, they're abnormal because of this heavy processing. And they really only function short term, which is fine and works well for liquid tumors, like I had said, but doesn't persist and allow the penetration of solid tumors or the elimination of chronic viral infections. So our technology focuses on the use of blood-forming or hematopoietic stem cells. So instead of harvesting and processing peripheral blood T cells, we harvest hematopoietic stem cells from individuals and then modify them and transplant them back into the patient that last. And this enables a lifetime effect. So a lifetime transplantation with a technology that will continuously and naturally produce these T cells in the body. So we're using patented technology, which we patented through UCLA, and proprietary technology, which we've developed in our labs, to perform gene therapy to introduce these CARs and T cell receptors into minimally processed hematopoietic stem cells, which then engraft in the bone marrow in a patient to provide unlimited production of these genetically reprogrammed killer T cells that are enabled to target a cancerous cell or a virus-infected cell. So being produced in the body instead of the laboratory, these cells don't suffer the abnormalities caused by the laboratory manipulation that results in their short-term lifespan. So our solution provides a permanent supply of functional killer T cells that are targeted against a specific type of cancer or a specific type of virus-infected cell, which overcomes the challenge of persistent tumors or persistent viral infection. So despite the current limitations in this approach with peripheral blood T cells, there have been only 4 FDA-cleared chimeric antigen receptors since 2017, but they are very successful in treating, again, those types of liquid cancers. The T cell gene therapy market is projected to reach $17.3 billion by 2027. So it's a huge market. And the CAR technology and the TCR technology are really one of the prime focuses of the cancer field right now in targeting and specifically tailoring therapies to a specific type of cancer. Our technology, we believe, will broaden this market for currently untreatable cancers and chronic viral infections. So, for instance, human papillomavirus associated cancers alone account for 5% of all cancers worldwide and are currently incurable once they're metastatic. And we're hoping that our technology is able to target that type of cancer. Our primary indication and what we've really laid the groundwork in and our proof of concept has been in the HIV field, where about 38 million people live with HIV infection, including 2.2 million in North America and Europe. And the average cost of treating these individuals, while the antiretroviral drugs that are available to these patients are effective in suppressing the virus, the patient needs to take the medication for the rest of their life. So all of these patients are really in need of a permanent cure solution to allow the clearance of the virus. So what sets us apart from the existing technologies is that we have patented and proprietary CAR T cell or CAR molecules and TCR molecules, and we have a method and delivery system through hematopoietic or blood forming stem cells. So this stem cell angle on the treatment strategy is really what we have the technology in. And we have a portfolio of patented and proprietary chimeric antigen receptors and T cell receptors. And we also have the expertise to create new chimeric antigen receptors and T cell receptors against any virus or viral associated cancers. And together, the founders have over 75 years of combined experience in the viral immunology field. And we've all received over $100 million in funding from numerous agencies. So we have over 10 recent publications in this new and novel technology summarizing these results. We've indicated that we've published that these hematopoietic stem cells can be genetically reprogrammed with an artificial antigen-specific receptor, like a CAR and or TCR, that can naturally develop into fully functional, mature immune cells in vivo. So we used two different model systems to look at this. We did it in humanized mice, and we did it in non-human primates. So this is an in vivo proof of concept that these cells can be modified and then fully developed into T cells that target and contain these artificial receptors and can target the genes of interest. In non-human primates, we observed safe, long-term, more than 2-year engraftment of pre-programmed CAR containing hematopoietic stem cells, indicating that this treatment is safe and non-toxic. So these long-term studies really provided this proof of concept. We observed successful development and wide distribution of these reprogrammed sites at multiple anatomic sites in both these non-human primates and in humanized mice, which target virally infected cells, indicating that these cells can develop and distribute in the body like natural T cells can. Now, these resultant cells respond like they should. They naturally should, recognizing and killing infected cells, virally infected cells in vivo, indicating that these gene-modified cells are fully functional. We showed that HIV-specific T cell receptor and CAR T cells can lower viral replication in vivo, indicating that they are efficacious in killing virally infected cells and in lowering the amount of virus in the body. So all this provides a proof of concept that this CAR stem cell gene therapy can be efficacious in vivo. So how our therapy works, how this technology works, involves taking the stem cells from a patient in the primary indication. Like I said, the furthest along would be with HIV, but this would also be either a patient with a certain type of malignancy or a certain type of chronic viral infection. We would isolate stem cells from this. We would introduce a specific chimeric antigen receptor that targets that infection or malignancy into the extracted stem cells, and then transplant these modified stem cells back into the patient. All of this would essentially be ideally an outpatient procedure and be minimally invasive in treating a patient. So these cells would then engraft in the patient and then would develop naturally into mature T cells and other types of immune cells that would have this chimeric antigen receptors on them that would target the antigen of interest and provide basically a lifelong supply of immune cells to keep the infection or the malignancy at bay. So the company, CDR3, is comprised of us 3 founders who are now advisors to CDR3. There's me, a professor of medicine. There's Otto Yang, who's our medical advisor, and his background is in infectious diseases. And there's Jerome Zach, the chair of microbiology and molecular genetics, whose background is in HIV and other types of immune therapies. This is led by Matthew Lawrence, our new CEO. CDR3 is a Delaware company, and we have an exclusive license of 2 issued patents and 1 provisional patent from UCLA. And what we're ideally looking to do is to be able to acquire funding to enable the technology to develop new targets, new therapeutic targets, and take this development and eventually cross that bridge to eventually put it into patients and commercialize the technology. I'm happy to take any questions at this point.